I didn't think I was going to have the other outline with me, but then I realized I could get it, so I was afraid I'd forgotten it. Well, I had forgotten it, but I was able to get it. So, I've talked to a couple of you since the last lecture, and I think that the message got delivered reasonably well, that signaling is really important when we think about cancer and we think about control and the things that cells have to manage. And so I'm going to finish up with a couple cancer-related things with respect to signaling today, and then we're going to turn our attention to metabolic energy. Okay? So, when we look at receptors, we've seen that problems with receptors can cause some severe problems for cells. So I described mutations that can happen to an important signaling protein known as SRC. And if those mutations prevented SRC from becoming phosphorylated, then SRC basically couldn't be turned off, and so it was continually signaling the cell to divide. We saw a similar thing with RAS, where RAS lost the ability to break down GTP, leaving it in the always-on state, and it was also causing a cell to continually divide. Not all mutations affect protein structure. So I'm going to talk about one mutation here that affects not the structure of the protein that's made, but the quantity of the protein that's made. So mutations that affect the transcriptional control region of a cell, like, for example, with the promoter, all right, can affect how much of a given protein is made. So the quantity of a protein that's made can also be a factor in the control process. So let's take a look at that. All right. So the receptor I want to talk about today is an epidermal growth factor receptor. It's called the human epidermal growth factor receptor 2, or as I will refer to it here, HER2. Now, HER2 is a relatively minor receptor as far as cells go. Okay? It's not made in very large quantities, and it goes in the cell membrane, and interestingly with HER2, it's actually able to form dimers in the absence of epidermal growth factor. So HER2 can form dimers in the absence of epidermal growth factor. Since HER2 isn't present very abundantly in the membrane, this doesn't happen very often, and the cell isn't stimulated to divide very often as a consequence of that. Mutations, however, that affect HER2, all right, cause more HER2 to be made, and so the more HER2 that is made, then what happens is that the cell is getting a signal more frequently, even though there's not an epidermal growth factor bound to it. So the amount of HER2 that's present is a very important factor. Mutations that increase the amount of HER2 in a cell are factors in breast cancer, they're factors in ovarian cancer, stomach cancer, uterine cancer, and the interesting thing about these is there's actually a pretty good treatment for it. The treatment for cancers that are caused by quantities of HER2 are to use what's called a monoclonal antibody. So a monoclonal antibody is a very specific antibody that recognizes some very specific structures of one thing and one thing only. The monoclonal antibody that is used to treat HER2-related cancers is known as Herceptin. Now, I'll tell you what it does in a second, but I want you to know that name first. Herceptin is a monoclonal antibody, okay, that binds to the HER2 receptor. All right? Now, I've described that if we have too much HER2 being made, what happens is those HER2s can actually form dimers with each other without epidermal growth factor being present, and the cell signals to divide. If you have too many of these, you have a problem. So what Herceptin does is it binds to one of the two units and prevents dimerization. Okay? So you treat a patient with Herceptin to stop that dimerization because the cell is making too many in the first place, which is why the cell is continually getting this signal to divide. And if you stop them from dimerizing, then what happens is the cancer is essentially stopped. Now, Herceptin is a very powerful treatment that has very little in the way of side effects because it's pretty specific for that HER2 antibody. Okay? Are there side effects? Yeah, there are side effects of any treatment. So I don't mean to say that it's absolutely absent of side effects. But the beauty of this is that it's very specific for that HER2 receptor. So one of the things that we're increasingly doing as we see cancers and we're treating cancers is we're characterizing what type of a cancer is it. It's very easy to screen and see, are you making too much HER2, for example? And if you have that situation going on, then that treatment becomes automatically prescribed for you. Okay, that makes sense? Some yeses. Okay, yes, Casey. So why doesn't HER2 dimerize as frequently when it's at normal levels? It's a good question. HER2 is not very abundant normally. It's relatively minor in the scheme of things. It's not too abundant. So the receptors just don't find each other. It's only when you make a lot of it that they start finding each other more frequently and then causing that signaling to happen. Okay? To a limited extent, they do find each other. I don't want to say they absolutely don't. Okay? And as long as they're finding each other occasionally and causing a little bit of division, cell division, it's not a problem. It's when there's too many of them and that signal is continually being made that that's a problem. Does that make sense? Okay. Other questions? Yeah. Is HER2 present what? In triple-negative breast cancer? I don't know. I don't know. Sorry. Yeah, question back there. Is HER2 a needed component for a cell to divide? Yes, HER2 can actually dimerize with an epidermal growth factor. It doesn't absolutely require it, but it can, and cells are making it for a reason. So I won't say that we can just throw it away, all right? But when we have too much of it being made, we want to basically slow that down, and that's what the Herceptin is doing. That's a good question. Would HER2 be more important than epidermal growth factor if you're trying to get a cell to divide? If you're talking about an individual cell, all right, on average, the epidermal growth factor is going to be really an important component for that cell to bind and get that signal. HER2 is a relatively minor component of that, so I won't say it's absolutely essential, no, all right, but it can help to stimulate a cell to divide. The problem with HER2, I want to point out, is mainly one of amplification. Just making too much of something that's not normally very abundant at all is the problem. Okay? So this is an example of something that's not a structural change to the protein, but rather an increase in the quantity of the protein that's being made. We could imagine that we could see problems if we had some proteins that were necessary to stop uncontrolled division. If they weren't being made at all, then that could also cause a cancer. And there are examples of those as well. Okay. And there's the dimerization. All right, I want to talk about one last set of mutations. This also affects structure, and this is a very interesting one, and it's particularly of interest for people who are from Oregon, for example. Okay, I'll tell you why. So this relates to a mutation that involves the sort of recombination of parts of chromosomes, and those parts of chromosomes contain part of proto-oncogenes. So the proto-oncogenes that are involved are called BCR, you see on the right, and ABL, ABL, which is shown on the left. Now these two are enzymes, are protein kinases, tyrosine protein kinases that are involved in signaling processes. And like you've seen of other tyrosine kinases, they can affect the decision of a cell to divide. So when they're active, they will cause a cell to divide. If they're not active, they won't cause the cell to divide. Okay? Now, the problem here is that when cells go through a process of division, there's a phenomenon called crossover or recombination that can occur, where parts of one strand or one chromosome can sort of swap with another. And when that happens, you get what's called crossover. This involves chromosomes 9 and 22, which is where these two genes are located. When these two guys cross over, you get something called a BCR-ABL fusion, meaning that the BCR, part of the BCR protein is joined to part of the ABL protein. Everybody with me? The part of the ABL protein contains the tyrosine kinase. Now, ABL is normally made in cells at a fairly low level, kind of like HER2, except it's not a receptor, it's just an internal tyrosine kinase. So it's made at fairly low levels. And at low levels, it functions properly in that signaling process, telling a cell when to divide or not activating a cell when it shouldn't divide. BCR, on the other hand, is made at higher levels in the cell. So what's fusing is part of BCR and the control region for BCR. So you got the control region for BCR, BCR, and ABL all linked together. That means that the amount of the fusion protein that's going to be made is the amount of BCR that would have been made if it were BCR by itself. The amount of the BCR-ABL fusion that will be made is the amount of the BCR that would have been made. And since BCR is made in a higher quantity than ABL, we start to see a problem, right? Yeah, repeat that one more time. So we're fusing the control region of BCR, the first coding region, the first part of the coding region of BCR, and we're linking those two things to the part of the coding region for ABL. So we make what's called a fusion protein, where the BCR coding is linked to the ABL coding. It's a brand new protein. That BCR-ABL didn't exist before then. We put those two pieces together. We've made a new protein, and that new protein is able to phosphorylate tyrosines. It participates in the signaling process. Since we have the control region for the BCR, it's the control region of BCR that determines how much of a protein that is going to be made. Okay? That fusion protein is going to be made at the level that BCR alone would have been made, and that's a high level. So now we've got a tyrosine kinase that's being made in high quantities. That's a brand new protein because it's this combination of these two, okay? And we have problems arising from that. Now, that's the bad news. The good news is that it's a tyrosine kinase. And people have asked the question, what if we used a tyrosine kinase inhibitor? Could we stop this signaling process and stop this cancer? And it turns out the answer to that is yes, and it's a very interesting yes. Because there's a drug called Gleevec. Okay? Get to it. There it is, Gleevec. All right? Gleevec is a tyrosine kinase inhibitor, and here's the really good part. It tends to prefer to bind to the BCR-ABL over other tyrosine kinases. That is, it will preferentially inhibit the BCR-ABL fusion over any other tyrosine kinase. Well, you couldn't ask for a much better magic bullet, folks. A magic bullet is something that you shoot and you only kill the cancer cells. And only these cells, by the way, it's known as chronic myelogenous leukemia, that people who have this fusion, those cells have a protein that no other cell has. So you treat this with a drug that targets that fusion protein and stops it. Now, the reason this is of interest to people in Oregon is there's a man named Brian Drucker up at OHSU who is the world's leading expert in this. He's actually the one who realized the value of doing this when nobody else was doing this. He's brought it forward, and he's actually, people are discussing, possibly up for the Nobel Prize in Medicine because of this discovery. This magic bullet that's used here, okay, has doubled the 5-year survival rate of people who have chronic myelogenous leukemia. Okay? Wonderful drug. And it has worked miracles for many people. So it's an incredible, incredible, powerful drug. Now, that's kind of convoluted. Does that make sense? Questions? How often does what? How often does the fusion occur? As often as a cancer occurs? So, yeah, it's not a common thing, but this type of leukemia is not unknown, certainly. It's not uncommon. So there's a decent number of people who have this fusion who are living very normal and active lives today because of that drug. Okay. A question somewhere? Yeah. So what's left, you mean back here? There? Yeah. So what you see is you sort of see a swapping that's happening, okay? So the BCR-ABL fusion occurs on that right chromosome. The left chromosome is still perfectly functional. So both chromosomes are actually active. They will replicate, all right? The problem is just you've created a new protein that is out of control. That's the problem. Okay, well, let's go through. I think that's the last of what I have to say there. I gave you a summary, so I'm not going to go through that, all right? But you can use that summary, hopefully, to help you understand many of the points that I've made there. Okay, let's stop there, and we'll move on to metabolic energy, okay? And we will stop this guy and move this over. Everybody see it okay? Okay. So we're getting ready to move into the study of metabolism. Metabolism is the study of the biochemical reactions that occur within cells. We're getting now down to that molecular level that we've been talking about. In order to understand reactions, we have to understand energy of reactions, and that's what this talk is about today. We're going to introduce some terminology to you first. Okay? I've been using these terms to some extent during the term. Anabolic and catabolic. Something that's anabolic is being made. So anabolic processes are processes in the cell where large molecules are being made from small molecules. Making of a protein from amino acids is an anabolic process. Okay? Breaking down glycogen to release glucose is a catabolic process. So catabolic processes are the opposite of anabolic processes. They convert large molecules into smaller molecules. Anabolic processes require energy. Anabolic processes require energy. Catabolic processes release energy. Okay? So the reason we break down glycogen is so we can get glucose, and glucose, when we break it down further, gives us energy. If we want to make a protein, we'll learn next term that we have to have energy in the form of GTP in order to make protein, starting with something small and building it bigger. So anabolic processes require energy. Catabolic processes release energy. Anabolic processes tend to be reductive. That is, reduction is involved. Usually. It doesn't happen with a protein, but in other processes it does. Anabolic processes tend to be reductive in nature. Catabolic processes tend to be oxidative in nature. So again, yin and yang, opposite things. Okay? So anabolism is the result of something being anabolic. So anabolism is what is happening when cells are making things, and catabolism is what's happening when cells are breaking down things. We're going to see examples of both of them before the term is over. Now you can see examples on the screen there where we have large molecules in blue on the right, fats, polysaccharides, and proteins, that are being broken down into their substituent components, fatty acids, glucose, amino acids, etc. And those themselves can be broken down into even smaller molecules. That whole process going on the right is catabolism. Process going on the left involves the synthesis of things. Okay? The synthesis of things. So from anabolism, we get proteins, we get nucleic acids, we get fats, and we get carbohydrates. Okay. Well, this is depicted a little bit differently here, but it's basically the same idea. We have building blocks on the top right that are made into macromolecules. That can be DNA, that can be proteins, that can be fats, a variety of things. Those are going to require energy. Those are anabolic processes. Those are reductive processes. The energy you can see is coming from ATP. ATP is being converted to ADP as we move from right to left, providing the energy to make those molecules. So ATP, we all know, is of course critical for cells to function, and this is one of the reasons why the body must have abundant sources of ATP. An absolutely amazing statistic to me is that every single day you make and break down your body's weight in ATP. Every single day. That's a lot of ATP, folks. That's a lot of ATP. Think about that. You make and break down your body's weight in ATP. So ATP is rotated. It's revolved through. Even if you have a fairly lazy day, you're doing that. Okay? On the bottom, how do we get ATP? Well, we get ATP from catabolic processes. Catabolism is the catabolic processes that generate ATP, and we're going to see a lot about that later this term and also the beginning of next term. Okay. If we step back a little bit and we look at this biologically from the perspective of ecosystems, for example, we realize, of course, that all of the biological energy that exists on the face of the earth essentially comes from the sun, ultimately. Comes from the sun. Photosynthesis, okay, is capturing photons and using the energy of those photons to make sugars, and those sugars are used to provide energy for everything else that happens on Earth. We don't want to mess with the photosynthesis cycle, because if we do, we're ultimately going to starve ourselves to death, because that's where all the energy comes from that feeds our systems. Glucose is used to make energy in the process of catabolism. Glucose is used to make glycogen in the process of anabolism. So glucose is very central to both of those processes. Okay. I said we had to talk about energy, so let's talk about energy. If we have a reaction, A goes to B, and I have a small a there. That could be, for example, two A's go to one B. Maybe the two join together to make something, right? So the small letters refer to numbers. All right? If I have that circumstance, the Gibbs free energy change, which is given by the delta G, the Gibbs free energy change for that process is given by the equation you see on the screen. Delta G is equal to delta G0 prime plus RT times the natural log, the quantity of B, or the concentration of B to the B power, divided by the concentration of A to the A power. Now, for most of the things we're going to do, we'll just consider that to be B's and A's, the small b's and small a's, as 1. So we'll just say the concentration of B over the concentration of A. And I'll even simplify that further for you in a minute. Okay? I'll talk about what delta G and what delta G0 prime are, because that's the most common thing that students confuse. You guys will make me very happy if on the exam you do not confuse those two. Okay? So I must take a little time to go through that with you. I may have a multi-component reaction. AA plus CC goes to BB plus DD. I could have 20 of these, right? For that, you can see the equation gets a little more complicated, right? Got good news for you. You're not going to work with complicated equations. Okay? We're going to use the bottom equation in a simple form. We're going to say delta G is equal to delta G0 prime plus RT times the natural log of the concentration of products, remembering that that term, of course, comes from all that other stuff. But you're not going to use all that other stuff. You're just going to remember it as concentration of products, divided by the concentration of reactants. That's simple. No exponents, nothing else. Concentration of products of reactants. So that concentration of products and reactants is critical. Remember when we talked about Henderson-Hasselbalch? We said Henderson-Hasselbalch equation, pH equals pKa plus the log of salt over acid, right? pH was a variable. pKa was a constant for a given acid. And the amount of salt and acid changed as the pH changed, right? So salt and acid were variables. pH was a variable. pKa was a constant. Does exactly what I've just shown you. Delta G is the change in Gibbs free energy for a reaction. It's a variable. Okay? Delta G0 prime is a constant for a reaction. And the concentration of reactants and products can vary in a reaction. So we have variables, just like we had the variables in the Henderson-Hasselbalch equation. So the question arises then, what's the difference between delta G and delta G0 prime? All right? Now I'm going to say something to you you've heard in every class, but I want to help you to internalize that a little bit. All right? Remember, I'm a strong believer in the practical use of mathematics and chemistry to solve problems. That's why we don't use calculators. Okay? The practical use here. All right. The delta G0 prime is a measure of the change in Gibbs free energy for a reaction under standard conditions. Under standard conditions. Okay? What are standard conditions? Well, we've got standard temperature, standard pressure, and concentration of everything at 1 molar. That's what standard conditions are. Now, biological systems tend not to like protons at 1 molar because that would be a pH of 0. Right? So we make a special delta G0. It's normally called delta G0. We make a special delta G0 called that delta G0 prime. The reason that prime is on there is we have pH 7. We don't have protons at 1 molar concentration. Okay? So that's what the prime on that delta G0 prime refers to. We're working in a biological system. If we did it at pH 0, we would have no reactions going because all the enzymes would be dead. Okay? So the delta G0 prime is a constant for a given reaction. And it's a constant because we have an exactly set of conditions that are there. 1 molar everything. We have 1 molar products and we have 1 molar reactants, right? When that happens, what is the value of the log term? 0, right? The log of 1 is 0. When that happens, delta G equals delta G0 prime. They're the same thing at that point. Now, are all reactions done at standard conditions? No, right? So what if we had twice as much products and reactants? Would we still have delta G equaling delta G0 prime? We would not. But could we calculate the delta G? We could. We would simply plug in that 2 to 1, take the natural log, multiply it times the temperature, and multiply it times the gas constant R, add it to the delta G0 prime for the standard conditions, and now we would know the delta G under these conditions. What I'm trying to show you is that delta G relates to any conditions, and it varies as we change those conditions. Delta G0 prime is a constant, just like pKa was a constant for a given acid-base or acid-salt system, right? So, reactions in our cells don't occur under standard conditions. Most reactions don't have everything at 1 molar, right? But we can use that information under those standard conditions to see how the actual delta G is for a reaction given the conditions that we have. Does that make sense? So the delta G is equal to the standard conditions plus that amount that comes from the variable part of the equation on the right. Don't confuse the two. Now, here's where people confuse the two. When I go right here for A and B at equilibrium, and by the way, I've said before, I'll say it again, equilibrium does not mean equal concentrations. Please get that out of your heads, okay? Chemistry should have pounded that in everybody's heads, and chemistry didn't do a very good job of that. Pound chemistry's head, okay? Equilibrium simply means concentration is not changing over time. We might have 10 times as much A as B. If the concentration is not changing over time, we've got equilibrium. If the delta G for this reaction is negative, the reaction is favored in the forward direction, meaning that A is being made into B. If the delta G for that reaction is positive, okay, then that means that the reverse reaction, B going to A, is favored. If the delta G is 0, that means the A and B going to B and B going to A are equally favored, and that's why the reaction is not changing in concentration over time. Notice, delta G is 0 at equilibrium. Right? That's not delta G 0 prime, right? Everybody's got that? Please plant that in your head. You'll make me very happy if you do that, okay? You really will. At delta G, the system is at delta G equals 0, it should say, the system is at equilibrium. Okay. That was quick. Oh, sorry, there we go. There we go. Oh, hold on here, sorry. There we go. All right. Another thing to consider, all right? Yeah, question. No. At delta G, not the system is equilibrium. No, it should say at delta G. In fact, I'll fix it on the screen so you can see it, okay? At delta G, go back. Okay. At delta G equals 0, that's what it should say, the system is at equilibrium. That is the correct statement of that right there. If I said delta G naught is equal to 0, what would that mean? Oh, damn it, I keep screwing up. All right. What would it mean if delta G 0 prime were 0? What would that mean? It what? It's just a constant for a reaction. But it's actually a special case of a constant for a reaction. What would it mean if I had a reaction? I think you talked about this in the recitations this week. What would it mean if I had delta G 0 prime equals 0 for the reaction at equilibrium? What would that mean? What could I say about that reaction when it's at equilibrium? What's that? Go ahead. It would mean that the equilibrium would have 1-to-1 ratio of products over reactants. There's an extra credit point. Okay? You would have equal concentrations of products and reactants. And that's the only time, okay, that you would have equal concentrations of products and reactants at equilibrium if the delta G 0 prime is 0. Okay? So don't confuse delta G 0 prime and delta G. Right? Delta G 0 prime is a constant. Okay? Delta G 0 prime has nothing to do with equilibrium. It'll affect equilibrium, but it has nothing to do with equilibrium. Yes? Yes, absolutely. So the question is, can equilibrium be when they're not at equal amounts? Absolutely. But I said under the special conditions where the delta G 0 prime is 0, then they will be. How do I know that? How do I know that? All right? Remember, delta G is equal to delta G 0 prime plus RT times natural log, right? If I say I'm at equilibrium, what's the value of delta G? 0. And I said the special condition here, I have delta G 0 prime is equal to 0. So 0 equals 0 plus the log term, right? What's the value of the log term? 0. And if the value of the log term is 0, how does the log term become 0? Equal concentrations of products and reactants, right? Make sense? That's the only time that happens. The only time that happens is then. So good question. Yeah? There are times, oh, yeah, absolutely. His question is, are there times when delta G is 0, but the delta G 0 prime is not 0? In fact, absolutely. Every reaction can go to equilibrium. So that means, and every reaction has a unique delta G 0 prime, and they're not usually 0, right? So absolutely. What would that mean? Let's say I'm at equilibrium, and I've got delta G 0 prime is plus 3. What's the value of the log term? 1,000? No. What's the value of the log term? It's minus 3, right? If I got a plus 3 and a minus 3, they're going to add up to 0, right? Now, I'll tell you something that you will commonly hear people do, and biochemists are really bad about this, and I'm probably bad about this too. is they will talk about delta G0 prime as if it indicates the favorableness of a reaction. And in a sense, it does. Okay? Let me see if I can show you an example here. Here's a reaction that happens in our muscle cells. It's catalyzed by an enzyme known as creatine kinase. And creatine kinase is really important. I'm going to take a minute to describe this reaction to you. Then I'm going to come back and explain to you how this favorability comes in. Creatine kinase catalyzes the reaction you see on the screen. Creatine plus ATP makes creatine phosphate plus ADP. This reaction has a delta G0 prime of 12 kilojoules per mole. It's positive value. Okay? If I want to have equilibrium, what do I have to have? 0 equals 12 plus minus 12, right? If the log term is negative, what does that mean about the ratio of products to reactants? Do I have more reactants or do I have more products? That ratio has to be less than 1. I have to have more reactants, right? In order for that log term to be negative, I've got to have more reactants than products, right? Everybody with me? More reactants. So, more reactants means more creatine, more ATP, right? What if I start with equal amounts of creatine, ATP, creatine phosphate, and ADP? What's the value of the delta G? I've told you that the delta G0 prime is 12. And I start with equal reactants of products and equal concentrations of products and reactants. What's the value of the log term? 0, right? 1 to 1, log of 1 is 0. So what's the value of the delta G? 12. What's the direction of the reaction? Leftwards. I'm going to make more creatine, right? And ATP both, right? So I'm favoring the reaction to the left. This is where I get into the favorability thing. If I start with equal concentrations of everything, and I have something that has a delta G0 prime, it's positive, the reaction is going to be favored to the left, and that's what people mean when they say that the delta G0 prime tells the favorability. What if the delta G0 prime were negative? It would mean that it would be favored to the right if I started with equal concentrations of everything, right? Everybody understand that? Now, you may not understand that. If you don't, see your TAs, come see me. I'll be happy to step you through it. Okay? I'm going through it kind of quick, and it's easier if you sit down with you and show it to you. So if you want to see that, just come see me, okay? Or your TAs. Now, this reaction's cool from a physiological perspective. This reaction is really important for us when we go sprinting. I go sprinting, what's happening? I take off and I go racing down this aisle right here, which I'm not going to do, okay? The first few steps, I've been sitting on my butt for some time. The first few steps I take, I got abundant ATP in my muscles, right? And my muscles go, hey, let's go running. And I go about 10 yards, and all of a sudden I've used a ton of my ATP in those muscles. And what's happening to my ATP concentration? Going down, right? Well, I need to keep running. I got a grizzly bear chasing me, guys. I need ATP. And I don't want to have to wait for something to kick in like a hormone system that might take a few seconds to start generating ATP, or a minute or so to generate ATP. I need ATP now. Let's look at this reaction. When I'm sitting around and I've got plenty of ATP, okay, I got plenty of ATP. Which direction is this reaction favored? Left or right? Right, right? If I have a lot of ATP, the system's going to move to the right. It means I got a lot of creatine phosphate. Creatine phosphate is a storage form. Before I start running, I got a lot of creatine phosphate right there. And I take off running, my ATP concentration starts to fall. What happens when my ATP concentration starts to fall in this equation? It starts moving to the left, right? And when it starts moving to the left, what do I make? ATP. This is an ATP backup for my muscles. So when I'm sitting around, I have a lot of creatine phosphate. And when I start running, I immediately start making more ATP. I don't have to wait for this to happen. This is happening as I'm running. That's pretty cool. Now, people get obsessed with this, and they do dumb things with it. They decide, okay, well, if this is really good for you, then I can be a better athlete if I take more creatine. And some people think they can do that. And some people do that. Right? Is that necessarily good for you? I would say probably not. Is it bad for you? I would say probably is, but then notice I said probably. My advice is don't mess with this equation. Okay? Let's imagine that you decided that I want to really boost my energy, so I'm going to go out on a sprint today. I'm going to go take a bunch of creatine, which people do. What's the concentration of creatine on the left on this equation? It's high, right? If I take a lot of creatine in the morning, I got a bunch of creatine sitting there, and I take off on my run. What happens to my concentration of ATP? Goes down. The equation wants to move to the left, but what's going to keep it from moving too far to the left? Too much creatine, right? Not the smartest move on earth. I just stopped this reaction from going to help me to do something. And people say, what if I took creatine phosphate? Okay? Well, you could try that, I suppose, but I wouldn't recommend it. But people do that all the time. Okay. That's just a word to the wise. All right. Here's what we're after, ATP. ATP is critical because with ATP, that's the gasoline that we use for cells, right? ATP is necessary. And the reason ATP has high energy is because if you look on the left, you see those 3 phosphates in ester bonds. Phosphates are negatively charged. Those negative charges repel each other. Those phosphates want to get away from each other, but you'll notice they're covalently bound to each other. The reason we get energy from hydrolysis of ATP is we're allowing the phosphates to go boing, flying away. Once you cut the covalent bond, you release energy. This is why. Okay? Okay. Oxidation, essential for energy. With oxidation, okay, we start with something that's highly reduced. The more we oxidize it, the more energy is released, which means that the thing is that is the most reduced has the most potential energy. On this figure, carbon dioxide on the right has no energy to provide for oxidation. That's why you see a 0 above it. That carbon on the left, that is bound to either other carbons or to hydrogens, has the most energy because it is the most reduced. Oxidation provides energy. Cells don't oxidize things by simply just burning them, because if they did, we would have flames and we would be on fire all the time. You hear about somebody who's really hot, right? It may have a different meaning than perhaps what this shows here. So instead of oxidizing things and producing flames and a lot of heat, we do produce some heat. Instead of producing the heat of a flame, for example, we transfer electrons to electron carriers. You see an electron carrier on the screen. I've talked about it before, NAD. NAD is the oxidized form of an electron carrier. NADH is the reduced form. The molecule on the left is reduced. That's known as glyceraldehyde 3-phosphate. You don't need to know that right now. The molecule on the right is oxidized because it has now become an acid. It's actually in the ester form with a phosphate attached to it. That's an oxidation to go from an aldehyde to an acid. The energy of that oxidation was released, and the cell used it to make NADH. Okay. There is the actual reduction reaction that occurs. No, you don't need to know those structures. But you can see this is nicotinamide, adenine, dinucleotide. That's what it's called. That's where NAD gets its name from. Okay? And the actual reduction is happening where those arrows are showing you right there. And again, you don't need to draw this or anything. I'm just showing you this for your information. Okay. I'll come back to that later. Come back to that later. Got several things I'll come back to later. But I thought we would go to this. Everybody's been wound up over the election, so I thought we'd do 2 songs today. Everybody in favor of that? First one's called O Delta G. Pretty easy to sing. O Delta G, the change in Gibbs free energy, can tell us if a process will advance. 'Cause if the value's less than naught, it translates back. Reverse reactions haven't got a chance. But when the sign is plus, it is the opposite. And then the backwards happens all the time. A factor is the standard gives free energy. So don't forget about the delta G naught prime. And I just realized one announcement I forgot to make is that I will do that session I promised you tomorrow night at 7 o'clock, ALS 4001. ALS 4000. I will videotape it, so if you're not there, that's fine. Okay, one more quickie to get out of the way of the next class. This one is the Muscle Energy Song. And for jumping, you need some energy. Quickly, the body stores it in the form of ATP. I can't sing this one. Backup should be needed. Reserves are there in wait. Creatine phosphate. Ready whenever, whatever, wanting to exercise. Steady as ever when whatever energy needs to rise. Reaction is exacting for leaping in the air. Myofibrils all contracted, using energy extracted from reactions that react in me, using ATP. See? All right.